Hi everyone, thank you so much for clicking onto the video. Here are today's learning objectives. So the first is to look at the platinum-2 complex cisplatin, which is used as an anti-cancer drug. And we're going to see how it prevents DNA replication in cancer cells by a ligand replacement reaction with DNA, in which a bond is formed between platinum and a nitrogen atom on guanine. We're also going to appreciate that society needs to assess the balance between the benefits and the adverse effects of drugs, such as the anti-cancer drug cisplatin. So by the end, we should be able to explain why cisplatin prevents DNA replication and explain why such drugs can have adverse effects. So now we're going to look at anti-cancer drugs. Cancers are actually cells which have just lost control over their own growth and replication, and they grow much faster than normal cells. However, there have been anti-cancer drugs which have been discovered. And we're going to look at one example, which is cisplatin. And this is one of the most successful cancer treatments that we found. So how it works is that firstly, it bonds to a strand of DNA and it distorts the shape. And this prevents replication of the cells. And so the cells we're stopping from replicating are these cancerous cells. And the molecule bonds to the nitrogen atoms, as we have a lone pair of electrons on the nitrogen, which can form a dative covalent bond with the platinum on two adjacent guanine bases on a strand of DNA. And the nitrogen atoms on the strand of DNA actually displace the chloride ions, which are already on the cisplatin, because they are better ligands. And this is a ligand substitution reaction. And cisplatin can also bind with adenine on a strand of DNA or across strands, but this is less likely to happen. So what we have is cisplatin, which has the formula platinum, and then we've got 2 chlorines, and then 2 ammonias. So how we bond it is we have some DNA. So we've got the double helix of DNA. And then say here in the middle, we have these 2 adjacent guanine bases. So we have a guanine here and a guanine here. And obviously the complementary base for a guanine would be a cytosine. So we'd have 2 cytosines over there. And then we'd also have hydrogen bonding between these 2 bases. And then we have this cisplatin, which comes along, and then a ligand substitution occurs, and so it bonds, the platinum bonds to the nitrogens of the guanine, and the nitrogens displace the chlorines. So then what we get is platinum, and then now it's bonded to these 2 guanine bases via the nitrogen on the guanine, and then we have still these NH3s. So this is our DNA strand. And the side effects of cisplatin is that firstly, we need to know that cisplatin can't distinguish between healthy or cancerous cells. And so that means that it'll bond to DNA in healthy cells as well as cancerous ones. And this can be a problem as some healthy cells replicate just as quickly as cancerous cells. And so they'll have a larger effect on the replication of these cells compared to cells which don't replicate as quickly as the cancerous ones. And so, for example, hair follicles actually replicate really, really quickly. And so the cisplatin kind of thinks of it as a cancer cell. And so it bonds to these healthy cells, these healthy hair follicle cells. And that's why patients tend to lose their hair during chemotherapy, which is a form of cancer treatment. And this is because if we've bonded this cisplatin to these healthy hair follicle cells, then it prevents the replication of these cells. So basically we stop the replication of hair cells, of growing hair. And so that's why patients tend to lose their hair during chemotherapy. And something also really important to remember is that if we have transplatin, which is cisplatin, but instead of these 2 groups being on the same side, they're on opposite sides. If we have transplatin, Cisplatin does not work as an anti-cancer drug.